Good morning, everybody. It is a great pleasure for me to introduce Dr. Bhattacharya for today's seminar on our 7th annual symposium GNE Myopathy meeting. Dr. Bhattacharya is currently a professor of biology at Ashoka University in Sonipat, India. He has trained in some of the most prestigious institutions and universities in the US, including the National Institute of Health and Harvard, and is also a renowned scientist in the field of genetics and therapeutics. Many of our patients and their family may also know his work promoting research to identify a cure for GNE myopathy through the charitable trust that he named Work Without GNE Myopathy India that he and his family have set up. Dr. Bhattacharya today will be presenting a talk entitled Development of a Therapy for GNE Myopathy, and we are looking forward to his presentation. I want to remind everybody to please mute your microphone at the bottom of your screen and to turn off the cameras so that we don't slow down the internet. And without any further ado, I'll let Dr. Bhattacharya introduce his work. Thank you so much.
Thank you very much. And I really would like to thank NDF for giving me this opportunity to talk about some of the ideas that we have and some of the problems that we could face in future as and when therapy comes out. So essentially, my talk will be mostly on what do the patient expect from drug developers, because the patient have some need. And I will give some idea about what is the current status and how the drugs are developed. And for GNE myopathy, what are the potential approaches that can be used for drug development, and what is happening in the world right now with respect to other genetic disorders, and how do we go forward. And then we'll talk about accessibility to drugs around the world. And we'll, in the end, I will try to suggest some ways by which, as and when the drugs are developed, how it could be made accessible to all the populations in the world. Basically, what do the patients need? Patients actually need drugs that can cure or reduce and block progression of the disease. The ultimate goal is to find a cure. And if we can't find a cure, at least we can reduce or block progression of the disease. And obviously, with respect to GNE myopathy, we need drugs that can regenerate damaged muscles, and drugs that are obviously accessible to everyone around the world. So the idea is that whatever damage has been done, if we can find drugs to regenerate that, if we can absolutely cure everything and bring back people to their earlier state, and finally, of all these drugs, if they're accessible to everybody in the world. Though there are two types of therapies that there for rare genetic disorders. Most of the therapies that are available currently require to be taken lifelong. In most of the time, these therapies actually help in slowing down the disease, and for a few years, or if for a decade or so, enhance the quality of life. And 99.9% of the therapies that are currently available do not cure it. Okay. And the second kind of therapies are the cure, or what we call one-shot or one-time treatment. And for example, gene therapy and bone marrow transplantations are examples of that kind. In these cases, the patients actually are fully recovered from the disease. So what are the various ways that we can actually develop drugs? And next few slides, I'll talk about that. And I'll just take you through simplified ways by which the different processes that can come into the picture, and there can be different targets where therapy can be targeted to. For example, the muscle cells are undergoing degeneration. Okay, so one of the ways to obviously to treat the disease would be to replace the bad muscle cells with the good muscle cells. Then the reason why the muscle cells are degenerating, because the DNA, there is a mutation, there is a change in the DNA, and we know that in GNE myopathy, GNE gene is mutated. So one thing we can do here is that we can actually either correct that mutation, or we can actually replace with a good gene which doesn't have a mutation. So that can happen at the DNA level itself. Now what happens is that DNA actually are actually made into or transformed into another set of molecules called RNA, and this is a process called transcription. We don't have to worry about it. These RNA are important molecules because they are the ones that finally help into making proteins. So we can actually treat at this level. So we can actually supplement with good RNA, good messenger RNA, so that this good messenger RNA can make the functional protein that is there. Okay, so one way to develop a treatment is to supplement with good messenger RNA, and that would lead to functional proteins, therapeutic proteins, and that would help us to find a cure for it. And then once the proteins are made, because of these changes in the sequence, because of the change in the DNA itself, the proteins quite often do not fold properly. Fold means the protein comes out as a single linear chain, is like a big string, and then generally in the cell, they kind of fold into a small compact structure, and that's how they are functional. And it is possible that mutation would cause this problem of folding. And because it doesn't fold properly, they are not fully functional. We know that in GNE, because of the mutation, they have a little bit of problem in producing enough quantities of sialic acid. So one way to treat here is by adding something called chaperones, which will actually help into fold the protein and make it functional. And then at the protein level, we can actually replace, we can add a purified protein from outside, which are fully functional proteins, and that these purified proteins can then replace the bad protein, and then that's how they can do it. And finally, the protein, though is not a fully functional, we can find something called modulators or small molecules, which can make the protein into fully functional one. They're like a drug molecule. They go and bind to this protein, though it's a mutated protein, and this protein, the small molecule that goes and binds, can help this protein to become fully functional. And then, finally, we have metabolites, in this case, sialic acid, and we can supplement with sialic acid. So these are overall, full steps, different steps where we can intervene and come up with a new drug. I will not go into details of most of this, what are the current effort goes into this area, but I will say that what I describe into the systems and many diseases that people are coming out with intervention at the different levels that we have. For example, there's a gene therapy that, as I mentioned, that where you can actually send a normal gene into it, a functional gene, and make it work. And there are different ways of sending the gene. There is a viral vectors, and there's something called non-viral delivery system they can be do. And then, of course, there are gene editing technologies that are coming up that can correct it. And we talked about replacing damaged muscle cells with a good one, and that can also be done using stem cells, which is the technology that is being developed. And there are two ways of doing stem cells. One, you take the stem cells out from the patient, correct the gene there, and send it back to it. And by that, you can actually have good stem cells, which will then lead to formation of new muscles. There is also a possibility that this whole process of taking stem cell, correcting maybe too much and may take too much of time and money. There's also possible that you can take stem cell from a healthy donor, and that stem cell can then be used for a patient. So you don't have to correct the DNA there. And then, of course, there are small chemicals. Like any other drugs, most of the drugs are small chemicals, and they can be used to support. And I described that in the previous slide. So what are the current efforts that are still going on? And majority of these efforts are actually towards enzymes and protein replacement therapies. There are a large number of diseases, lysosomal storage disorder, many blood disorders, where pure enzymes are given and pure protein molecules are given, and that's how you actually help in restoring some of the functions of it. And this is a therapy that is going on for the last 25 years, and it does help patients for some time, but tremendously expensive. There is also RNA therapy that I talked about, and for many diseases like Duchenne muscular dystrophy, there's something called exon skipping. That is a small ways by which you can sort of get rid of the mutated part of the DNA, and you generate a messenger RNA protein which is free of that mutated section. And that allows some functional activity to be restored. And this is also very popular in many genetic disorders. So what are the approved therapies for a large number, for a few diseases that has come out and already in the market? There's already a gene therapy for SMA1 that has come out. There's exon skipping therapy that I talked about, how small RNA molecules can help to remove that mutated part of the DNA, and that is available for both DMD and SMA. And these are all have been approved and in the market. A large number of enzymes and proteins, which I talked about, are already available. And for Fabry disease, chemical chaperones is already available in the market. And there is a small molecule called ataluren, and that is something called it's a very specific, so that the defective part, the defective change is kind of suppressed, and so that you can actually make a functional protein. So those are available. So there is a lot of development that has happened where you have you have the approved therapies that are in the market, and we should look towards some of these development because they can give us a way by which we can be successful. So what are the requirements for a new drug discovery? This process is intricate, process is complicated, and it requires, of course, a lot of money. And it requires a basic understanding of the biology. Why, for example, a mutation in the DNA, in GNE gene causes muscle defect. Now, interestingly, the GNE gene is needed every place in the body. It's needed in the brain, it's needed in the liver, it's needed in kidney or anywhere else. But why it affects only skeletal muscles? That's something we still don't understand. And we need to have a large number of different kinds of scientists come together and clinicians come together to understand this. If we understand this, we will be able to figure out a way to solve the problem, way to find drugs. And we need technological platforms. How do we go and find a drug? So we need something called a large-scale drug screening. And today, the approach is very simple. You make thousands of different types of chemicals. And then you set up something called a screening technique, where you screen every molecule to see whether it is performing like a drug or not. So that something is very, very useful to figure it out or find new drugs for any disease. And that's kind of platform we need. Then we need a lot of biologics. For example, gene and cell therapy system. We need a vector that will take things in. We need stem cells. We need delivery systems to deliver them in the right place. And that happens to be a major problem. Not only that, we need also manufacturing capacity. Not only that, we have to show that in a few patients, you can show it, you can make it in a lab, and then show that it works. But then if you want to take it out in the market or you distribute to all the patients, you need to have a manufacturing, you need to have quality control of those manufactured products, etc. So all these technology platforms are needed to develop drugs. And that's why majority of drug development process happens in big companies, because they have these technological platforms. Then, of course, we need regulatory approval and large-scale production capability. So drug development process is not simple. All these things need to come together, and we need a lot of collaborations with a large number of different experts. So what is the major problem in GNE myopathy? Why we are not able to still find a drug for it? So some of the issues that for which we still don't have an answer is that why, as I pointed out, why skeletal muscles get affected. Okay, other muscles like heart muscles or breathing muscles don't get affected, or other tissues don't get affected, where salicylic acid is required everywhere in the body. And we also know that if you somehow stop production of salicylic acid completely, okay, then nobody survives. That survival is not there. So salicylic acid is needed even for survival. But in GNE myopathy, it is probably reduced slightly, and that's what causing. But why it's causing skeletal muscle, we still don't. Now, we also find that age of onset is not same, anywhere between 20 to 40, 45. And also severity of the disease not same. Why some people have more severe disease, some people. If we answer these questions, then it will be much more easier for us to find drugs. And this whole process, how this pathogenesis, how muscle defect is seen, there are two competing hypotheses that is there. One is that sialation is less, or what we call hyposialation, that salicylic acid production is less, because of that it is happening. The other hypothesis is that GNE is not only involved with salicylic acid production, it also does something else, and we still not clear about what it does. So, and that's why I said that our major handicap has been so far is that we don't understand the basic biology of this disease and how this mutation or changes GNE causing the muscle defect that we see. And I don't want to go into detail of it. There's a whole lot of different ways a scientist looks at this mutation effect, what we call phenotype, with respect to the genotype, which is the mutation. And the biology has a multiple ways of looking at it, and we think that there's a whole different mechanisms, or any one of them or multiple mechanisms may be involved in it. I've only talked about protein folding. There are others in it, including fact that how proteins get degraded. Every proteins get degraded inside cell at a certain rate. And maybe the mutation causes, a faster rate of degradation, etc. So all this process can be studied, and we do have tools to study all these things. So it is necessary that we really invest time and effort to understand why this GNE mutation shows the kind of phenotype that we have. So what has happened so far in GNE myopathy? There have been 2 clinical trials so far, and you are all aware of it: ultragenic salicylic acid supplementation therapy, which has been withdrawn at the phase 3 level, and Manac supplementation therapy, which is phase 3 is about to start, and phase 1 result, which was published, and a little bit of phase 2, which has been published, and shows that essentially what you see is that reduction in progression of the disease, but mostly in the upper limbs. So it's a it's not the ideal case, but it's definitely some kind of a looking forward that we can do. So what do we do for GNE myopathy? I think we can look at drugs that can probably be developed even without understanding the pathological mechanism. And I think we should use those approaches to do that. If you don't understand how biology of it, are there ways by which you can actually develop drugs? There can be. For example, we can do gene editing. We know that is the mutation in the gene that is causing it. So we can definitely correct that mutation, or we can send gene into it, good gene replacement. We can do that. And for these kind of methods, you don't need to know why there is a damage that is happening, what is the mechanism. Then we can, of course, look for small molecules. And I talked about how you can actually do screening using thousands of molecules. And there, all that you need to do is set up a screening procedure. For example, if you want to increase the sialic acid level in cells where there is a genetic mutation, you can use that as a way of screening molecules. Any molecule that will do that, you can then take it forward to see whether it does have activity or not. And then stem cells is another way that you can do it, for which again you don't need to understand the basic biology of this process. But sometimes basic biology, even in these cases, are necessary. But we can definitely try working with methods and approaches for drug development for which we don't need to understand why GNE mutation causes the phenotype that we see. So is it possible to make use of drugs that are being developed or already approved for other diseases? For example, there are some studies in GNE myopathy that shows that mitochondria is damaged. Mitochondria is the cell's powerhouse. It utilizes oxygen, generates energy, and they're very, very important for this thing. There are approved drugs that can enhance mitochondrial health, and it may be useful to actually look at some of these to see whether they have any positive effect or not. There's some experiments done in animals that shows that they may have some of these positive. And as I said, that folding is definitely seems to be one way that mutation can cause problems in GNE protein, and loss of ciliation can be because of misfolding. The inclusion bodies that we see, the rim vacuoles that one sees in the damaged muscles, they may be because of the floating. And there are large number of chemical chaperones. These are the molecules that help to fold, have been approved by FDA, and those can be tried out in a system to check whether they have an activity or not. And then again, there are large number of gene therapy vectors are right now either development or already in phase 1, phase 2 trial. And many of these actually target muscle cells. And because these vectors actually have propensity to get into muscle cells. So we can definitely try some of these vectors which have already been used for delivery of a good gene into muscles. We can definitely try for GNE. So we can make use of the technology and systems that are being developed for other diseases and see whether they work for us. What should be the way forward? I think the way forward should be to develop drugs that are not require basic understanding. I think we should go straight for those things. As I pointed out, that drug screening, stem cells, gene replacement, gene editing, that we need to do, and that can be started right away because you don't need to know biology for that. Then, of course, we need to also do basic studies on understanding how gene defect causes muscle defect, and that is very, very important. So we need to learn more about muscle cell biology and how GNE mutation affect those things. So those could be two approaches that can use. One can take a bit longer time, like understanding basic biology. The other can be really started off. So what are we doing it with our World Without GNE Myopathy? So we had set up about a year and a half back what we call a virtual center for drug development. Our CDD, or the Center for Drug Development, was set up to support development of a cure for GNE myopathy. So we wanted to support to do that. And our approach is slightly quite different. It is that we only plan to give strategic funding, or we have given only strategic funding, very small amount of funds for initiating research, and particularly in developing platform technologies, helping people to develop platform technologies in this direction. But we are also trying to sign getting people together, like scientific expertise that is available in India, as well as the infrastructure, scientific infrastructure that's available in India, for getting them interested in our project, that is finding cure for GNE myopathy, and then help them to write proposals which we can send to different funding agencies. India has a few funding agencies. They could support some of these projects. And we have been doing that, and we are hoping that some of these projects will get funded so that we can start it. Because government has much more money that any of us can actually do. We also want to utilize resources, infrastructure, drug manufacturing capability of public and privately supported organizations and companies. Many companies have the capability, so we want to utilize them, talk to them, partner with them, and to develop this. And then finally, we are very interested in patient-centric approach and participate in drug discovery. In India, the patients don't participate in drug discovery process. Many of the decision-making system don't have patients, but we are pushing for patients to be included in this thing. So some of the research that we have supported or supporting, or the research projects that we have submitted for funding, are the development of gene therapy system for GNE myopathy, using both viral and non-viral vector technologies. We are banking on non-viral vector technologies because they have much less side effects and much easier to make it, and therefore it will be much more cheaper if it really works. We're also trying to work out human cellular models for GNE myopathy because of the problems with animal models. If we can get human cellular models and the data from there, if it's acceptable to our drug development drugs equivalent to FDA, what we call CDSCO, then that will be really nice. And our third approach, which is we are pushing quite for, is the identification of potential drug-like molecules by using an intelligent design-based approach, as well as large-scale screening, which I talked about, that you can actually do screening where you can find potential drugs without actually knowing much about the biology of the system. So I come to the an important aspects of my talk, and which I think is very, very important, is that where does a rare genetic disorder stand in the context of in India and other developing countries? And there are problems, serious problems, which the developed countries cannot understand. And for example, awareness about the disease among lay public, doctors, decision makers, and scientists is very generally low. Large number of doctors don't know anything. Of course, public you don't expect. Most of the decision makers don't know. Scientists are also there. And one of the things we are trying to do is to make particularly scientists and clinicians aware of it. And we also trying, there are issues there that are very, very important. We are trying to find ways by which we can solve it. And it would require participation of people from social sciences and other disciplines. For example, there are cultural and social problems associated with genetic disorders. They are not accepted by the society. And so large number of patients not go for diagnosis or will not go and see a clinician, and family won't talk about it. So we just don't know how many patients are there. And that's an important problem. There are also very few clinical centers where diagnosis can be done. And of course, our pharma companies are not really interested in developing drugs in this, and partly because of the small population that is there. And when treatments are available somewhere in the world, costs in these countries are prohibitive. And this is point I will come to that. And one of the problems that people can't access any of the drugs that's developed elsewhere is that there's no health insurance or government-supported scheme that will pay for the treatment. So a poor patient, even a middle-class Indian, cannot afford most of the treatment that are currently available anywhere. And there's many of the developing countries do not have orphan disease policies, so there is no context on which government takes decision. So I just want to come out because something that many people in the US and Europe may not be aware of, they may be aware that the treatments are cost are very high, but they're so high that, and with no insurance, because many cases the insurance pays for it. With no insurance, how you can pay for it, how anybody can pay for it. So these are some of the treatment that has come out in recent times, and you can see that even a steroid treatment is a few $3,500 per year per patient. A steroid is a very, very cheap molecule to synthesize, but they are not available in it. And you can see that what could be the potential cost of it. In fact, for example, for Spinraza, the cost of treatment per patient per year is more than the salary of families in over their lifetime. And if you really look at the one-shot cure, those are the ones that you have to take every year. If you look at the one-shot cure, the spinal muscular atrophy gene therapy is about $2 million for a shot. There's no way anybody can give that. Okay, even bone marrow transplantation is not that easy for it. So the issue is that how do you make drugs accessible to everyone? And that's a very important one. That whenever we think of development drugs, we also should think of whether it will reach everybody in this world, and how to do that. That we should think right from the beginning when the drugs are being developed. There are many ways to do that. If the cost of drugs are subsidized by government or charitable organizations, if let's say 99% paid by charitable organization, 1% paid by government, still it is possible to do that, though many poor people in developing countries will not be. Drugs are distributed on no profit basis. There is no reason why the cost of these drugs should be that high. The manufacturing cost or whatever the cost you can take it, this should not be that expensive. There's no reason why it should be that expensive, because the cost is being calculated based on a different parameter, and that doesn't take into account even a small profit margin of this production cost, profit margin, R&D cost. If we put all of this together, still the cost would be probably 1/10 or 1/100th of what is being charged. Now, it is also possible that drugs can be manufactured in a developing country under license, okay, with low fees. And since the cost of production is very low in these countries, this can be made cheap. And one of the examples is anti-HIV drugs. The India started manufacturing and distributed at 1/100th of the cost to everywhere in Africa. So African HIV control was mostly because of the drugs being supplied from India at an extremely low cost, $1 a day. Okay, and a similarly large amount of COVID vaccines are being also planned to be manufactured in India, and they're going to make it with $10 for a COVID vaccine, and the ones that are made in India. If the technology is available freely for rare disease drugs, somebody can definitely make it at a very low cost. So I end my talk with some of the conclusion that we can come from this, or some of the points that we can take from here. There is only one potential therapy in the horizon for GNE myopathy right now, which is the MANAC. It too is not ideal, but it is definitely something if nothing is there. And we need to increase our effort towards understanding pathobiology of the disease, thereby increasing, of course, the prospect of finding an effective therapy, which is very, very important. But in the process, meanwhile, we should keep working towards developing gene and cell-based therapies, small molecule screens that do not require an understanding of the disease. So we, one hand, study the basic biology, other hand, we continue where we don't need to understand basic biology. We also need to find ways to make drugs accessible to everyone in the world. And I believe that patient organizations such as NDF, WWGM could bring all stakeholders in the quest for finding a cure available to everyone. And our motto is that the cure should be available to everyone. Thank you very much for your attention.
Thank you. This was a great presentation. We have a few questions that are coming in, and I want to start with a question of my own, actually. I would like to know your comments about the function of GNE and whether there could be other potential players that play a role in the disease, and what's your thought about the hyposylation, the misfolding, and other potential mechanisms?
So I think GNE probably has other functions on it. If you, there's some work that has been done where there are GNE is part of a network of proteins. That means in the cell, what happens is the proteins don't exist in isolation. The proteins actually are, they all interact with each other. It's like a small society where they interact, and these interactions are very important to get the whole function there. And what can happen is that the mutations could actually affect these interaction processes. Some they may increase and some they may decrease. What happens, what kind of interactions and their role in different function has not been really worked out. And this is one area that we need to work out to see what else that GNE is doing, what things can happen if some of these interactions are not allowed to happen. And they can happen. For example, one of the things could happen is that GNE mutation in GNE could cause GNE to not localize into a place in the cell where it normally goes. It something called mislocalized. It goes to a different place. And then if it goes to a different place, it does different things there and all. And there are other proteins that help this localization, and that's the reason why it is. So there are large number of different possibilities to do that. And I think the point that I said, that most important thing to understand why skeletal muscles get damaged in GNE, because there are other genes in the sialic acid biosynthetic pathway. There, the mutation does not cause skeletal muscle defect.
We have a question from Mangesh. Mangesh is asking, from the presentation we have from Dr. Kelly Crow, we know that the lab is working on identifying a biomarker for GNE myopathy gene therapy. Are there similar biomarkers needed for some of the other potential solutions you mentioned? Are any other labs working on these biomarkers that you know of?
Yeah, I think biomarkers are important because the screening strategy that you can use can be used, it would require some kind of biomarkers to do that. One of the simplest biomarkers, we still don't know whether that's the most relevant for the disease or not, is the lower level of sialation that you or hypersialation that it is there. If we use hypersialation as a biomarker, then you can actually use it to screen molecules to see whether the molecules can actually increase the level of sialic acid. Now, I want to become a bit more technical here to see that. So, mutation in the GNE gene could affect, as I said, two things. It could affect its other properties other than sialic acid, and of course, it does affect siality. There is a change in amount of sialic acid that is formed. So it is possible that if you can bring up the level of sialic acid using a drug, then it may be able to restore other functions that are there. So any biomarker and what we call a surrogate biomarker, that is a biomarker, this is not directly involved but indirectly involved, could help in doing it. But I agree with Mangesh, and what I'll say is that we do need biomarkers and what are for any kind of drug discovery process. But currently, as I said, that hypersialation is a reasonably good marker that one can use because we think that that marker could also affect others.
Excellent answer. And Tara is asking, how close are we to supplementation with messenger RNA for us? And is any group working on this?
Not for GNE myopathy, but there are two, three diseases work is going on. In fact, there are 2 or 3 different companies that are working on it. You all are aware of this company called Moderna, which is the front runner in making messenger RNA vaccine for coronavirus. Moderna has a small team that works on rare genetic disorders, and they are using messenger RNA to do that. They have the technology platform for production of messenger RNA, and they're doing it. In fact, even Ultragenics is also interested, and they have a collaboration with another company that's interested that have some of the technology for messenger RNA treatment. So this is one area that I think we should very seriously consider.
Do you know of what would be the expected delivery efficacy to muscle, considering that we have to target virtually every single muscle if we use a messenger RNA or any other recombinant protein, for instance?
So many gene therapy vectors are pretty good in delivering things to muscle. We have seen from large number of studies done in other muscle disorders, DMD, LGMD, etc., etc. Even Pompe's disease, where the effect is on muscles. So there are vectors, for example, AAV9, which is, and people have modified AAV9 that goes into muscles pretty well. And also same way, messenger RNAs are generally delivered through something called a non-viral packaging system. So there are chemical packaging systems, and they can be modified to target it to different tissues to do that. And this is a technology that's really developing very fast. And one of the reasons why it is developing fast is also the fact that antisense oligonucleotide, that is exon skipping oligonucleotides that are used for treating some of the diseases like DMD or SMA, are also packaged into some of these non-viral packaging systems and that are used for targeting. So I think that there's enough knowledge in this whole world now, and many companies have the technology for targeting to muscles.
Along the same line, Tara is asking, what is the purified protein that you mentioned in your talk, and is it available? And what would be the mode of delivery?
See, what happens is that almost all lysosomal storage disorders and many of the blood disorders, you make large quantities of this purified protein, something that you can do it in test tube. Large vats, you can make these purified proteins, you modify them so that they are stable, they don't degrade, and then you can modify them in a way that they're taken up by cells very easily and survive within the cells. These are pure protein made outside human body, outside systems. These are made in factories, and these proteins then go there. I'm not sure. There's a possibility that nobody has ever tried that, whether we can make GNE protein large quantities and inject it and see whether it works. That's worth trying.
Yeah, I want to shift a little bit the question to a different topic, which is the stigma associated with knowing or being a carrier or having a genetic disorder. Can you perhaps comment on what is the strategy, at least in India? We know that India is a country that puts a lot of pressure on castes, and what is the strategy to move forward and to overcome this problem?
See, we don't know. I mean, I think we don't know. We are talking to other people to do that, looking at it. In fact, today there's some discussions that I'm sort of involved where people are talking about ways by which societal issues can be circumvented through a very directed education system, very directed kind of promotional material to do that. So the issue is simply that if a family knows that they have someone with a hereditary disease, then what happens is that if a son has a hereditary disease, they will think that if people come to know of it, their daughters will never get married. Okay, and because of that, they will never talk about it at all, never get diagnosed. And what we need to educate people to see that it's important for these boys to do that, and we can do testing for others to see whether they have the same mutation, etc. So all these things need to be projected in a way which will make people aware of the consequences of it, make them safer, and show them that it's more important to come out clearly rather than hiding the whole thing. And this is a very complex issue which requires participation of social scientists, psychologists, other things, and we need to do that in a large thing. My university, we have a big social science group. They are actually interested in some of these issues, and we are going to take it. Right now, we really don't know how to go about it.
We have a question that just came in from Tammy, and Tammy is saying, my son is 16 with GNE myopathy. He also has a platelet disorder. His hematologist believes that the low platelets are due to low sialic acid. Would it be easy to see if a drug worked on him by watching the platelet count? His platelets are around 40,000. Now I know there are at least 4 other GNE patients that also have a problem of this type. Thank you so much.
Okay. Um, I'm not very sure whether a defect in the GNE gene would cause a platelet functions. That nobody has actually looked at what else it may cause problems. You know, originally people thought that it could also affect the heart muscles, etc. But we know very sure that smooth muscles, which is the heart and other muscles, are not affected so much as the skeletal muscles are affected. So it's not very clear what it happens. But you know, we also don't know, for example, a severe form of hypercoagulation, whether that would cause problems in platelet or not. That's something we don't know. There is a range of phenotype you see in GNE myopathy, and this range, you could have other associated problems that come in. We know so little about the biology of sialic acid that whereas sialic acid is needed by every cell type in the body, that it's difficult to say right now. You said that 4 patients already have this platelet problems on it. This is worth actually looking at it and see what can be done. I mean, I can have a look at what kind of levels of sialic acid biosynthetic genes are there in platelet and how that can get affected. A quick look, we can do it and then come back to you.
Yes, actually, there are some more comments where Tammy says, I can send the article, and it was published in the Blood Journal. And it's definitely worth, Tammy, if you don't mind, to reach to us and send us the article, and we'll be more than happy to forward it to Apple.
That will be nice.
Yes.
That will be nice. I would like to look at it.
I have a comment on a question actually on the making any drug available to people around the world independently of the social economic status of the world. We know that India is a large producer of a manufacturer of many of the drugs. I'm not sure about AAV vector, but I know that India is used extensively as a source of chemical manufacture for the world. Would that be any way that we can convince governments to make those drugs available in developing countries by subsidizing the cost of the drug and obviously making it cheaper and affordable to people that don't live with the social medicine, or they don't have the same economical status as the European community or the North America? And is there any plan of at least from the government in India and your position to try to get to that place, making it available?
Yeah, that's a good point. This is something that I pointed out, that there are manufacturing places, and thanks to COVID, the new technologies are coming in. For example, I think the messenger RNA, the Moderna product will also be manufactured by one of the, at least partly manufactured by one of Indian companies. So they have the capabilities to manufacture and manufacture it very cheap. Once we have a molecule, this is something, once we have a molecule, we would like to take it up, talk to them. I have a brief talk with one of them, and I think they will be all supporting it as long as cost for production comes out from there. They may not be looking for big profit out of it. Then we need an international consortium that will make the technology available to them, and they will be then, and the government that can subsidize partly, and then of course, if they are willing to take it up. And I have a feeling that they will be willing to take it up to do that, because this anti-HIV was one of the projects that our government and the African governments, they collaborated with the drug company, and they managed to get this done. I think that's definitely a possibility, and something that I also mentioned in my talk. One of my slides had some of these things. And once we have a molecule, and I think we'll go all out to see whether it is produced at a cheaper rate, provided it doesn't cost us millions of dollars to get that technology.
Yeah, I agree. And please do let us know if us as a community can do something to, when we get to the point of producing it and making it available to everyone, if there is anything that we can do and make our voice heard.
Sure.
Okay, I think we don't have any more questions coming in. I want to thank Dr. Bhattacharya for this beautiful presentation and for introducing his thoughts and for helping us navigating this series online. I will thank you everybody for staying with us. Please stay safe, stay healthy, and reach out to NDF if there is anything that we can do. Thank you.
Thank you.